Thank you very much for that kind introduction. I'm honored to have been asked to participate in the first VLB International Brewing Web Conference. Today I'm going to review a project that probed the chemical makeup of NABs and how this influences flavor and preferences of North American consumers. This study was performed in collaboration with the VLB while I was a postdoctoral researcher at the University of California, Davis in Dr. Hildegard Heyman's lab. This work was published in August, and you can see the cover art for this right here. At the start of this project, we had three main questions, which we probed with three different techniques. First was, how do the aroma, taste, and mouthfeel profiles of already commercially produced NABs, or non-alcoholic beers, vary? And to do this, or to probe this, we used sensory analysis. The next question was, what are the aromas, tastes, and mouthfeels Northern California consumers were most or least satisfied with in non-alcoholic beer, or NAB? And to probe this, we performed consumer analysis. And finally, were there any non-volatile or volatile constituents in the different NABs, which could be related to the flavor characteristics associated with either the least or most preferred NABs for Northern California consumers. And to probe this, we did some chemical analysis. So why non-alcoholic beer? Generally, we were interested in these questions because there seems to be a lot of interest in developing new products for this space currently. And the reason for this is because non-drinkers, millennials, and Generation Z consumers are driving increased demand for healthier beverage options. To cater to this demand, NAB globally is projected to double by 2024. Historically in the US, though, however, this category has been extremely small. However, there's growing interest, and you can see that there's a lot of growth potential in this market, at least in the US. So that leads to the first question. How much do the flavor profiles in different NABs vary already that are commercially produced? And while there has been a few studies evaluating the flavor characteristics of specific styles, such as Pilsner type NABs, and how this impacts consumer preference, there hasn't been a broad study to look at all the styles that exist within this market. And so we wanted to start with that. Therefore, we obtained 42 NABs from 6 different countries. It's important for me to point out, because of the propensity of non-alcoholic beer, aroma and flavor to be impacted by staling reactions, we obtained a very fresh product. Most of the NABs were either less than 3 months old at the time of the evaluation, or evaluated before the best buy date on the package. As you can see here, we evaluated a number of different styles of non-alcoholic beer. For those familiar with this market, we made no specific distinction between alcohol-free beers. However, the ABWs were considered later in chemical analysis. Another interesting point is that for one of the lagers, we tested 2 different package types. That was Lager 5, and we looked at this product in both bottle and can. Similarly, we also tested 2 other products as controls, a commercial seltzer and a commercial soda. While I won't really talk about these products today, look for future work on why we included these products in our analysis. The trained panel was made up of 11 judges who had self-identified that they consumed beer on a regular basis, as well as had a high preference for beer. You can see here the panel performing consensus training. And so what we did during the training sessions, which were 5-hour-long sessions, was the panel used consensus to generate different terms which describe the aroma, taste, and mouthfeels of the different non-alcoholic beers. Then after each session, I attempted to make a reference by chopping up an apple or squeezing an orange which best described the standard or term that the panelists were using. Now, while the panelists could use their own vocabulary to describe the samples to help generate terms and guide the samples in term generation, we also gave them the second edition of the beer flavor map by Draft Lab. Because of the number of different NAB styles that were evaluated, the panelists identified 41 different terms, and reference standards were developed to describe all of these different aroma, taste, and mouthfeels that the panelists came up with. You can see the standards that were developed to describe the terms here. Now, to make the evaluation less fatiguing on the judges, DA was performed only on terms which the panelists decided that they could evaluate over all of the different samples. Terms like bitterness, salty, sour, things that would be at least varying in all the samples. Terms that were more unique to specific samples, we decided to use a replicated kata approach. And these are the terms here in dark red and dark green. So something like grape nuts or bananas, which might only be in a handful of samples instead of all 42, were performed using this approach. So here's just a little bit more about the unique features of our sensory approach. To get the panelists ready for testing, we performed warm-ups with the standards that I just showed, and this was done in a randomized fashion. So before each testing session, the panelists had to come in and evaluate these standards, randomly identify these standards. And once they had performed this, they could go into the testing session. During testing, 60 mils of sample was evaluated after 30 minutes after 30 minutes after opening the package. And the sample was at about 14 degrees Celsius. The panelists tasted in these Belgian taster glasses, shown up in this right corner. And you can see the testing environment here, which was performed in individual ventilated and light isolating tasting booths under red light so that the panelists could not identify the color of the samples. Judges were instructed to first evaluate the aroma of the samples, then to evaluate the taste and mouthfeels of the samples, and then to cleanse their palates between each sample with carbonated pectin water. A 30-second break was forced between samples, and then between 6 samples, because there was 11 on the panel, there was a forced 3-minute break. Sensory data was collected on Chromebooks and Qualtrics. The panelists were instructed to evaluate, again, the aromas first and then the taste and mouthfeels. They were also instructed to evaluate the descriptive analysis terms first and then check all that apply for the unique terms that describe that sample. And again, this is an example of that testing space where we had the line scale for descriptive analysis and the check all that apply boxes. So let's jump into some data analysis. So we used 3-way ANOVA with the factors panelists, replication, and sample to process the descriptive analysis data. When accounting for significant panelists and replication effects, all the terms used for descriptive analysis were found to be significantly different for at least one of the samples. I've included the results for overall aroma intensity here. And what you can see is that with the Fisher's mean comparisons, that there are significant differences amongst the samples for this attribute. To compare the significance or evaluate the significance of the replicated kata data, we use Cochrane's Q-test for each given replication. And again, we found significant sample differences for all the terms evaluated using the replicated kata approach. To compare the kata data and the DA data, the results for each of the kata terms were summed over the 3 replications. So if a term had been selected by all 11 judges over all 3 replications, this would have resulted in a max total of 33 check marks or points, let's say, for that given term for a given sample. And so this sort of resulted in a pseudo-scalar value. As an example here is the banana term that we performed with kata. What you can see is wheat 1 and wheat 3 were very, very high in this term, essentially 23 and 27, very close to 33, similar for lager 5. And so these samples were, again, had this unique character, and it didn't necessarily make sense to scale this attribute over the whole panel. And the replicated kata approach seemed to work very well. To generate a visual map of the sensory profiles of the different products, we then perform principal component analysis on the correlation matrix of the least squared means of the DA panel, as well as the sums for the different kata terms, again, from the train panel. We can see the first two principal components here accounted for about 40% of the variation amongst the samples. It's important for me to point out here that the DA terms for the aromas in taste and mouthfeels are in light red and light green respectively, while the kata terms are dark red for aroma and dark green for taste and mouthfeel. So for example, thin is a kata term, and astringency and drying is a term that was a DA term, whereas for aroma, warty was a DA term, and Cheerios was a kata term. Generally, the terms on the right side of the plot were those such as warty, Cheerios, dried yeast, grape nuts, and these were driven more by malt and yeast, whereas the terms on the left side, such as tropical, citrus, stone fruit, herbal, and hop aroma, were those that were descriptive of flavors more driven by botanicals, such as hops or citrus juice in the case of the Radlers. If I now add in the different products into the PCA space, we can see that generally the products on the right side of the plot, again, were those that were malty and warty in character, and were the wheat styles as well as the lager styles. And you can see that there's a lot more of these styles than what's on the left side of the plot. The products on the left side of the plot, again, were the more herbal, citrusy products, and these were the pale ales, India pale ales, or IPAs, and hop waters. Further, you can see that there's segmentation as well as you move up and down in PC2. The wheats and lagers on the bottom right side of the plot, such as lager 2, wheat 4, and lager 6, were more malty, stale, thin, and bitter, whereas the lagers that were in the upper part of the quadrant, such as wheat 3, wheat 1, and lager 5 can, were sweeter and had more fruit-like aromas. In comparison, if we look at the left side of the plot now, the pale ales, India pale ales, and hop waters on the bottom left side of the plot, such as pale ale 6 or hop water 1 or IPA 3, had aromas which were more herbal and black and tea in character, and these products were more bitter. Whereas the pale ales, India pale ales, and IPAs that were on the top left part of the biplot, such as pale ale 2, hop water 2, IPA 1, were more citrus, tropical, and floral in character. So it's apparent that lager type NABs still make up a majority of the market. It's clear that these results show that NABs can and are being designed to have a wide range of different aroma, taste, and mouthfeel profiles. These results were then used to select 12 of the most diverse flavor profiles for consumer analysis. I've highlighted these samples in blue here. So that leads us to our second question. What are the aromas, tastes, and mouthfeels consumers are most and or least satisfied with in NAB? While there has been a few studies evaluating how the flavor characteristics of specific styles, again, Pilsner type, impact consumer preference, there's not any information on the more broad drivers of preference or flavor, how these very broad flavor profiles might influence the NAB market. So based on this, we decided to take these very diverse products and see what consumers would think of them. So prior to the pandemic, in November of last year, we had 144 consumers come in and taste these 12 different samples over the course of a Saturday and Sunday, and over 12 different sessions. Again, the testing environment that we used for consumer analysis was identical pretty much to the train panel environment. For the consumer panel, 144 consumers were recruited. However, only the data for consumers who were under 21 did not self-identify as an American consumer or had not been a U.S. resident for greater than 5 years were removed for the panel. Because again, we wanted to evaluate only the what American consumers thought of these different products, as this had not been done before. This resulted in a final data set generated by 129 consumers, 66 males and 63 females. The outcomes for the demographic surveys are shown below, and essentially they showed that the panel was made up of young, educated beer drinkers. The data collection for consumer analysis was broken into 4 parts. First, the consumers tasted through 6 samples. For each sample, they were instructed to rate their preference using a 9-point hedonic scale shown here. And the first attribute that they rated was the overall impression of the product. Then they were instructed just to look at the how much they liked, or consider how much they like the aroma of the product, and then consider how much they like the taste and mouthfeel of the product. After they had evaluated the sample with those 3 scales, they were asked a couple more questions seen here. And between each sample, there was a forced 30-second break, during which they were instructed, again, like the train panelists, to rinse their palate with a 0.1% carbonated pectin water. After they made it through the first 6 samples, consumers were then asked to fill out a demographic survey. They then evaluated another 6 samples in an identical way to the first set. And finally, they had an additional survey at the end of the panel that gauged their preferences around purchasing, consuming, and enjoying non-alcoholic beer. So let's dive into the data again. Again, using ANOVA and the Fisher's mean comparisons, we can see that there was significant differences in the hedonic likings between the samples for overall liking, aroma liking, and taste and mouthfeel liking. Here you can see the least squared means for these hedonic likings sorted by overall liking. What we see is at the bottom, the most preferred products, which were the Radler 1 and Hopwater 2. At the top were the least preferred products, which were the India Pale Ale 3 and Pale Ale 1. To explore the relationships between the consumer preferences and the sensory data, we used external preference mapping. External preference mapping was performed with the consumer hedonic data, as well as the PCA factor scores from the PCA made with all of the data from the trained sensory panel, but only using the PCA factors for the 12 samples evaluated during the consumer analysis to generate a vector model. These results are then displayed as a contour plot, shown here, representing the percentage of satisfied consumers, where satisfaction was defined as the percentage of consumers giving a hedonic rating higher than the mean hedonic rating for the set of the 12 samples evaluated during consumer analysis. The regions in dark blue represented a low percentage of satisfied consumers, whereas the dark red regions represent a higher percentage of consumer satisfaction. We then can take that contour plot and overlay it or underlay it on the PCA plot, where the 12 PCA scores were taken from. And again, I showed you this plot before. Again, this plot was made with all the data from the trained sensory panel. In doing this, we can now see what aroma, taste, and mouthfeel characteristics are driving consumer preferences, as well as how consumers might perceive the other samples that were in the trained panel, but were not able to be included in the consumer panel. Again, the products more towards the top in this orange space were the most preferred products, while the products towards the bottom in this blue region were the least preferred products. What we see is that the consumers were least satisfied with L6 and W4 in the bottom right hand portion of the pie plot. These products were more thin and bitter and characterized by more malty, skunk, stale aromas. However, a higher percentage of consumers were satisfied with Wheat 1, Lager 5, and Lager 9. Comparatively, these NABs were more sweet, cloying, and thick, and had aroma profiles that were more honey-like and fruity. Consumers were also least satisfied with Pale Ale 1 and IPA 3 in the bottom left-hand portion of the pie plot. Again, these NABs had aroma profiles that were more herbal in character and had taste and mouthfeel profiles which were more bitter, astringent, drying, and thin. In comparison, a higher amount of consumers were satisfied with IPA 1 and Hop Water 2. These products were less bitter and had aroma profiles which were more citrusy, tropical, and floral in character. Finally, way up the top was the citrusy sweet Radler 1, and this was the most preferred product by the consumers. Therefore, there seems to be general flavor profiles which the Northern California consumers were preferring. Just briefly, the additional survey that we collected at the end of the consumer session also tells an interesting story and supports these findings. For example, the top 4 reasons for wanting to try NAB were health, a thirst-quenching alternative to alcohol, abstaining for alcohol for a while, and flavor. These results support current market research trends and indicate that lifestyle trends might be driving consumers to search for alternative products, but also highlight that flavor is a critical consideration for consumers and their interest in trying NAB. The styles the consumers said they were most interested in trying were fruit beers, India Pale Ales, then Pale Ales. Given that 43% of the consumers in our consumer panel had not tried NAB prior to the survey, this supports that there's room to grow and develop this category in the US. So that leads to our final question. What are the chemical constituents leading to the most and least preferred flavors, and how can we use some production techniques to influence these factors? To do this, we used a host of volatile and non-volatile methods to break down the composition of the different NABs. For the volatile chemistry, this was performed at the VLB, and they used a number of techniques to evaluate the hop aroma compounds, staling aldehydes, dimethyl sulfide, as well as some other lower boiling point compounds, such as fermentation esters and alcohols. For the non-volatile chemistry, the more basic beer quality factors, such as alcohol by weight, pH, bitterness units, carbon dioxide, turbidity, were performed in Dr. Glenn Fox's lab, which is formerly Dr. Charlie Bamforth's lab at University of California, Davis, while again, the more sophisticated analysis, the hop acids, were evaluated at the VLB. So again, we performed external preference mapping, but this time we related the aroma hedonic liking scores of the consumers to the PCA scores for the 12 samples used for consumer analysis for from a PCA made with only the trained panel aroma data, as well as with the volatile data. Again, the most preferred samples are lying in the orange region, while the least preferred samples are lying in the blue region. What we see is a couple of general stories. The first is that, similar to previous research, that the least preferred NABs, shown here on the left, were characterized by malty stale aromas, which were associated with increase in staling aldehydes, such as trans-2-nonenal, while increases in fermentation esters here, as well as higher alcohols, seem to be associated with the more preferable flavors of the fruity lager styles. To produce more fruity NABs, a couple of strategies have been suggested. One technique that is gaining popularity because of their potential in alcohol-free beers is to use non-Saccharomyces yeasts, and there's evidence that some of these yeast strains lead to higher ester concentrations, resulting in fruitier non-alcoholic beers. This paper by Dr. Bellet is a great resource on this topic. Another technique is to blend in normal beer with a dealcoholized product to the legally allowable limit. And while this wouldn't be an approach that you could use in alcohol-free beer, again, this paper by Schmetzel is a great resource on this topic. The next story is that terpenes, esters, and aldehydes are the compounds associated with aroma profiles of the highly preferred botanically driven non-alcoholic beers, such as the Radler 1, which was blended with lemon juice, or the dry hop products Hopwater 2 and IPA 1. These volatiles were also significantly negative correlated with warty, malty, stale aromas, indicating that they mask or suppress these aromas. However, there were some botanical volatiles that were leading to more herbal characteristics as well. And these were associated with hexenol and pentanol, and increasing as you were moving up, and seen more in Pale Ale 1 or IPA 3. Obviously, juice and dry hop additions are leading to NABs with more preferred aroma profiles. Dry hopping is currently a popular technique being used by brewers to impart hoppy aroma while mitigating hoppy bitterness. And while there are a number of different dry hopping techniques which can influence dry hop aroma quality, based on previous research, dry hopping rate is one of the simpler ways. To estimate the dry hopping rate in the different non-alcoholic beers, we used humulinone concentrations. This is because humulinone concentrations are really only in appreciable amounts in beers that have been dry hopped. Interestingly, what we saw is that 48% of the NABs in this study contained humulinones. Therefore, the relationship which we found in previous research, which related humulinone concentration to dry hopping rate, was used to approximate the dry hopping rate of the different NABs in this study using humulinone concentration. Here you can see the overall liking of the different samples as a function of the estimated dry hopping rate. And what we can see is that, in agreement to the past study, is that the aroma profiles of the NABs with an estimated dry hopping rate of around 600 to 800 were the most preferred by both the trained and consumer panels, whereas those that increased over this rate were starting to become less preferred. And we think that this might mean that there's an optimal rate of around 600 to 800 grams per hectoliter, and that rates over 800 grams per hectoliter may be leading to the extraction of volatiles such as hexenol or pentanol, which are leading to these herbal characteristics. We then performed external preference mapping again, but this time we related the taste and mouthfeel hedonic liking scores of the consumers to the PCA scores for the samples used for consumer analysis from a PCA made with just the trained panel taste and mouthfeel data, as well as the non-volatile data. Again, there were also clear relationships between the analytical factors driving the differences in the taste and mouthfeel profiles and consumer liking. For example, the NABs that consumers were least satisfied with, up towards the top left, were those such as India Pale Ale 3 and Pale Ale 1, which had high sensorial bitterness, which was highly correlated with BUs and moderately correlated with high concentrations of isoalpha acid content. In comparison, the consumers were more satisfied with NABs more towards the right, such as Wheat 1 and Radler 1. These were sweeter and had higher residual extracts. Consumers were also more satisfied with NABs, such as Hopwater 2 and Lager 6, which had higher carbon dioxide concentrations, making them fizzier and more tingling. To investigate the main drivers of bitterness in NABs, we performed multiple linear regression analysis using all the non-volatile factors. The best fitted model showed that bitterness units and residual extract had the most significant impact on bitterness intensity and perception. Not surprisingly, residual extract was found to have a suppressive effect on the resulting bitterness, while the bitterness units had an additive effect. In NABs, the bitterness unit was really driven or was a function of isoalpha acid content. And again, this is the main, has been identified previously as the main driver of bitterness in beer, and results due to an isomerized reaction of alpha acids in the kettle. As mentioned previously, dry hopping could be a way to add hop aroma while mitigating hop bitterness, but dry hopping also adds humulinones, which are about 2/3 as bitter as isoalpha acids and should be considered on the overall bitterness profile of the product. In general, more bitter beer styles, such as the India Pale Ales, tend to have higher residual extracts to make them more palatable. However, in this study, the IPA styles had very, very low residual extracts. You can see that in blue here. And what we saw with these samples that had high bitterness units but low residual extracts was that they were very bitter and lingering. We can see the suppressive effect of residual extract here with IPA 1. It had a similar BU to IPA 3, but 3 times as much residual extract. Therefore, it added a bit of sweetness to this product, and this product had a more balanced taste profile, which consumers were more satisfied with, seen here in the green. So again, you can see that the consumer taste liking increased as more residual extract helped balance the profile of this NAB. However, an important factor to consider when increasing the residual extract is that this also increases the caloric content of the product. For example, seen on the bottom, Pale Ale 1 and IPA 3 are estimated to be about 15 and 25 kilocalorie per 12-ounce serving, respectively, while IPA 1 is about 76 kilocalorie per 12-ounce serving. The products that the consumers were most satisfied with were Radler 1 and Hopwater 2. These products also had very different residual extracts and caloric content. Radler 1 was very sweet and had a high residual extract, resulting in about 127 kilocalorie per 12-ounce serving, whereas the Hopwater 2, had a medium-low sweetness and had a residual extract of about 0.27 weight by weight percent, and had and this resulted in it having 0 kilocalorie per 12-ounce serving. Hop water, too, seemed to contain some form of alternative sweetener because it had some perceivable sweetness and such a low residual extract. Therefore, given that health and well-being are some of the main factors influencing consumers to choose NABs as an alternative beverage, this is evidence that the use of alternative natural sweeteners could also help to lead to more balanced taste profiles in these products, while also mitigating the caloric content. So with that, I would like to thank you all for tuning in today. I realize that I just covered a lot of information in a short amount of time. However, I have some good news. As I mentioned before, this work has been published in the open access journal, ACS Omega. Therefore, anyone interested in this topic further or is seeking more details should check this publication out. It's also at this time I would like to recognize the teams that helped to make this work happen. We send our extreme thanks and appreciation to all the anonymous brewers who donated fresh product to this study. You know who you are, and without such widespread support from the industry, studies like this are simply not possible. All the sensory work was performed in Dr. Hildegard Heyman's lab at the University of California, Davis with her amazing team. I would personally like to recognize Kay and Annegret for all their help, as well as all the other individuals listed here who helped to support and carry out the study. Similarly, all the more basic chemistry on the NABs was carried out in Dr. Glenn Fox's lab at the University of California, Davis. Again, I would like to thank Joe Williams and Kelly Scott for their support on this. The more sophisticated chemical analysis was performed at the VLB in the Research Institute for Beer and Beverage Analysis under the supervision of Dr. Niels Retberg. Once more, I would like to thank Johanna, Christian, Laura, and Jorg, as well as Niels for their support in this project. Finally, although I do not technically start my new fellowship until January, I would like to recognize and thank the Alexander von Humboldt Foundation for their support. Without this support, it's likely I would not have had the opportunity to share this work with you all today. With that, I would love to take any questions that you may have.